Hi, this is Menaka. I'm doing my research in Sastra Deemed University, Tamil Nadu, India. So now I'm going to present my poster in the title of ImmFinder, fully connected neural network approach for predicting the immune genes in livestock. So when coming into the introduction part, basically the classification of immune gene from non-immune gene in cattle is very important to understand their immune mechanism and their association with disease resistant, right? Because some cattle, like Indian cattle, have inherently resistant to multiple diseases and they have a very good immune mechanisms compared to other cattle like exotic cattle, hybrid and all. So, but traditional method like manual curation to extract the immune genes from other genes, it's very often time consuming and labor intensive. But there is database called INNADP database is available, but in that only innate immune genes are deposited, but there is no information about the other immune genes like adaptive immune genes and all. Therefore, we develop a model called ImmFinder, which is based on the framework of multimodal fully connected neural network that we designed to classify immune genes by integrating genomic and transcriptomic data sets. So the fully connected neural network is nothing but type of a neural network, but in this, every neurons are very interconnected. So this model integrate feature from different data types and capture complex and non-linear relationships. So the modalities we also call it as data set. So each modalities is processed through separate instance, and then we concatenate these two modalities, and then finally we do the classification. So a type of a joint learning from multiple modalities. So this type of model compared to traditional, it's very effective for capturing cross-model interaction in biological data and also improve the accuracy of the model. So when coming to the workflow, first we prepare the data set. So we chose two different data set for building the generating model. The one is genomic variant data, another one is gene expression data. So the genomic variant data we did by comparing the reference genome which is deposited in NCPA. The one is Taurus reference genome, another one is Indicus reference genome. We compare both and by using the GSLN and CD and identify the genomic variant. This ended up with insertion, deletion, and substitution. And another hand is gene expression data. We particularly collected expression data for infected and non-infected cases of cholera from inside from GU. This ended up with 7 different GSE IDs. Then we filter these based on a log to fold changes. If it is greater than 2, it means upregulation, and less than or minus 2, it means downregulation. Then we did data pre-processing. In that, we remove the redundancy and input missing values using Python. And then we classify the genes in each data set. So here we used INNADP database to retrieve the innate immune genes, and we used Python script to extract other immune genes based on a keyword-based search, like T cell, B cell, lymphocyte, cytokine. These type of keywords we use to extract other type of immune genes. So here we classify our data set into two different things. One is immune, another one is non. Then we balancing the class distribution to balance true positive and true negative is very important to prevent the bias over the particular class. So we used 10 different undersampling techniques to balancing the classes. So then we used two dense layer with 8 and 4 neurons for each data set. We also incorporate VLU activation function, L2 regularization, and drop rate of 10%. Then we concatenate these two modalities and then we incorporate one dense layer with two neurons and one output layer with one neuron. We also incorporate sigmoid activation function for binary classification. So this can classify if a particular gene is immuno. So in that we data set we split into three different types: 60% training, 20% validation, 20% test. So the training was conducted over 50 epoch with a batch size of 32. We also used Adam plus optimizer and early stop to prevent the overfitting. Finally, we evaluate the model using accuracy, precision, recall, and difference score. So when coming to the result part, our model achieved 85% of accuracy over our training test and validation set with the F1 score, precision, and recall of 0.85. So which confirming our model reliability and balanced performance. So this plot indicate the model accuracy over epoch. After ninth epoch, we observe the stable accuracy. This is the another plot represent the model loss. At the same ninth epoch, we observe the stable loss value of 0.05. So this indicate model maintain the generalizability and capture the significant patterns. So from ROC curve, we obtain the ANC value of 0.93. So this suggests model robustness to classify the immune genes from non-immune genes. In overall, our model is completely based on multimodal deep learning architecture to classify the immune genes from non-immune genes. So this model is particularly helpful to the researcher who are working into understanding the cattle immune mechanism. So if they have a long list of genes that are having a genomic variant or gene expression information, our model helps to quickly identify the immune genes from non-immune genes. So this further helps to fostering their downstream process like identify their role and their linking to the disease resistant traits and all. So further it supports to breeding decision and livestock development. So I conclude my talk with acknowledgement. Dr. Thank ICARE NASF for funding, Sastra Team University for research facilities, and MNCSB COSI ISCB for the opportunity. Thank you.